one of the reasons why Alzheimer's is really difficult to be treated is because it's not easy to be diagnosed at early stages. So what we're trying to understand is how is the pathology developing at the very early stages of Alzheimer's, so that we can develop some new diagnostic tool or treatments to diagnose the disease early and then treat it early. So the idea here was to identify the very specific neuronal subpopulations that are initially lost at early stages of Alzheimer, and understand why these populations, what makes them different from the other neuronal subpopulation, and which brings them to being vulnerable and eventually die. So we used, first of all, the main question was, how do we visualize these cells? Because there has been other works, for example, using single nuclear RNA sequencing that are very high throughput. So you can analyze almost all of the cells that are in the brain, and you can find the populations that are lost. However, with the single nuclear RNA sequencing, you lose the actual protein expression and you lose the spatial informations. Other techniques that were more traditional used normal immunofluorescence or immunohistochemistry, which however are very low throughput, so you only are able to analyze 1 or 2 markers at the same time. So in our technique, which is imaging mass cytometry, we put together, let's say, the spatial information, protein expression, and the higher throughput of markers analysis, so kind of joining somehow with pros and cons and limitations, but trying to put together the high throughput of single nuclear RNA sequencing but protein expression of immunostaining. And so the first question was, how do we visualize the majority of neuronal subpopulation in the brain, and then using that to identify which of these subpopulations are lost. And so imaging mass cytometry is really good because it allows you to stain up to 35 antibodies at the same time. So we could not only look at neuronal subpopulations, but then we could also stain for protein misfolding, which is obviously something that is very important pathology hallmark in Alzheimer's disease, which is again something that, for example, cannot be done in single nuclear RNA sequencing. And so the first big step was to try to optimize all of these antibody, and obviously we're studying we're conducting our research in post-mortem human brain, so it's also pretty challenging because the quality of the tissue is not always great and very variable. So anyways, after a couple of years, I managed to optimize a pretty good amount of antibodies, so we did manage to conduct the first analysis. And I'm now in the process of analyzing all of the data, which are, they seem to be quite interesting. And so we found some neuronal subpopulations that are selectively lost. And these are seems to be the same that have been previously identified in other studies. So this is quite good because it means that, our results are replicable based on previous results. And we also looked at how selective loss is correlated with misfolded protein accumulation. It seems like we see an intracellular accumulation of beta amyloids, but I don't want to go too much into these results because we're still in the way of confirming them with other techniques. So, but for now, it seems really exciting, and we're hoping to then be able to publish these not only as like interesting results on which to base further studies, but also as a tool that other people can use, because obviously these would be kind of like the first data set of so many protein expression markers on a human postmortem tissue, so it hopefully would be good for other people to use as well. So ideally, in the future, if we are able to develop better brain imaging techniques, we will be able to monitor the activity of the first neurons that are affected in Alzheimer, and that would be a great tool for diagnosing the disease at very early stages. And then At the same time, knowing which are the populations that are affected and why, we will be able to develop drugs that could target specifically the cell population and specifically the whatever molecular mechanism that is affecting their survival. So it's a great project for both developing a future diagnostic tool, but also drug target.